spoke in Portuguese.
Thomi understand Italian, Portuguese, Spanish.
Yes, true. Sorry. You speak a lot of language.
My Portuguese is unfortunately very, very poor. Very, very poor. Italian would be easier.
So we're live now, and what started, I would like to say good afternoon for everybody, and welcome to another session of the Human Awareness. Thank you for the participation, and especially thanks to Dr. Thomi Brunner invitation to talk about one of his main interests and seminal contribution to the understanding of molecular, biochemical, and cell biological process involved in immunopathological diseases and cancer development. And today he will be talking about extra-adrenal glucocorticoid synthesis at the epithelium and its role in inflammation and cancer. Dr. Thomi Brunner is the chair of biochemical pharmacology at the Department of Biology at the University of Konstanz in Germany. His lab to describe synthesis in a non-identifying tumor in humans with implications for through the synthesis of bioactive glucocorticoids in colon-dense. He also described how defective local steroidogenesis may contribute to the pathogenesis of allergic inflammation. All of his work has leads to 167 publications so far, obviously, with almost 14,000 citations and 8 index of 47. But after doing this more formal presentation, I also would like to give my personal testimony about Dr. Brunner's in Konstanz. I had the luck to spend 4 months there from November last year. And besides running a very productive lab, Dr. Brunner has the most amazing group of people, very clever, very hardworking people that are all testaments on what kind of research it is. And we also have many people here today in the audience. The ones that were here earlier could see how very happy people were to see Dr. Brunner. And I am too, I share in this feeling. Very excited to hear your talk today. So thank you again, and you have the floor.
Thank you very much, Bruno, for this super nice introduction. I'm flushing. You cannot see my face turning red, but anyway. I have to first of all apologize that last time I could not meet the appointment and give the seminar. Yeah, it was a bit of a hard time, and I'm glad I could get the chance to repeat this. As I was saying before, maybe some of you have heard this, some not. I'm currently at the conference at this lake here in the Swiss Alps. So hopefully the Wi-Fi and the internet connection is stable enough that we go through the presentation without troubles. And so this is what I want to talk today about. And first of all, this here, what you see is the University of Konstanz. It's a very young university. It's only about 50 years old. But as you can see, nicely located at the Lake of Konstanz. So we have actually just a short walk down to the lake. And what I really want to talk today is a long-term interest we have on the synthesis of immunoregulatory glucocorticoids. You all know glucocorticoids, I'm pretty sure, but not in the adrenals, but at so-called extra-adrenal sites, and how they contribute to homeostasis, immune homeostasis at the epithelial surfaces, and how they regulate also inflammation during tumor development. So this is an immunology seminar, so I don't have to convince you that the immune system is extremely important to regulate lots of processes in our body. Of course, the immune system is involved in the defense against pathogens like bacteria, viruses, but also parasites. At the same time, and now I have to, sorry, my screen is too small, so I have to reduce the videos, otherwise I cannot see the screen. Sorry. So, of course, this is a very, very important immune defense mechanism. It protects our body from potential pathogens. And at the same time, and I think there is more and more evidence for this, the immune system also defends our body against altered cells, altered cells, like, for example, tumor cells. So it's involved also in the surveillance of the body for developing tumor cells so that they cannot not grow uncontrolled. But a major problem is really that there is a very, very minor difference between tumor cells, tumor tissue, and normal tissue. And there is always this balance between, fighting tumor cells, controlling tumor cells, and at the same time not harming normal tissue cells, which could eventually lead to autoimmune diseases. So why is this actually relevant? Because former times, maybe we thought, tumor cells grow and they grow and grow, and at one point they're going to need and require blood vessels because they have an increased demand on oxygen and glucose and so on. But it's now increasingly clear that tumors are composed of very many different cell types, and a lot of them are actually immune cells. And whereas some of them might actually fight the tumor, others might even contribute to tumor development. They might just bring the right conditions into the tumor so that the blood vessels can grow, that the tumor can actually grow. So this is a very, very complex tumor microenvironment, and this is what I want to talk today in this presentation. So, for example, when we have a look at colorectal tumors, and this is a type of tumors where this has been extensively investigated, then we see actually that immune cells that infiltrate these tumors, they really have a major contribution to the growth and development of this tumor. And in my opinion, one of the very, very important publications that initially really confirmed this is the one by Gallo and colleagues in Paris. This paper was published in Science. There have subsequently other publications which investigated this specific aspect. And what they did is they were pathologists. They had so-called tumor microarrays. So they had stamps of tumors that were composed in a block, and then they could stain lots of tumors with, for example, the same antibody. And they just, in an unbiased manner, they looked at the infiltration of immune cells into the tumor. And you see here an example. So this is a colorectal tumor, and all these brown spots here, these are, in this case, T cells that infiltrate these tumors. And they statistically analyzed the tumors, how many T cells infiltrate the center of the tumor or the invasive margin. And they looked whether, for example, tumors that have a lot of T cells, they behave differently than those that have a little T cells. And what they found is what you see here, that those that have a lot of T cells, the patients survived much, much better than those that had little T cells. So what does this tell us? Obviously, the T cells that infiltrate the tumor, they control the growth of the tumor. That is kind of a tumor immune surveillance that helps the patient actually to overcome the tumor and survive. But it probably tells us also that tumors that have the capacity to suppress the immune system, as indicated here, they probably have an advantage. This is what we wanted to investigate. And I think there is increasingly evidence that tumors have developed lots of strategies to escape the control by the immune system, to escape destruction by immune cells. So, for example, many tumors have MHC downregulated. They just are not very good in presenting antigen. At the same time, of course, many tumor antigens are, or there are actually very, very few tumor specific antigens, and most antigens tumors express, the immune system is actually toleranced against. And also many tumor cells, they release, for example, immunoregulatory cytokines like TGF-beta, like IL-10, or express so-called immune checkpoint molecules, like, for example, PD-1 ligand that suppresses anti-tumor T cells, for example. And so often they even express molecules like Fas ligand or trail, which can even induce cytotoxicity in the immune cells and kill them. And how important this has been actually recognized by developing therapies where these so-called immune checkpoints are inhibited. So, for example, there are antibodies against PD-1 ligand or PD-1 receptor, and they would kind of neutralize this inhibitory effect tumor cells may have on the T cells. And how successful this is illustrated here. This Kaplan-Meier survival plot where patients, in this case, non-small cell lung cancer cells, or patients, sorry, have been treated with durvalumab, that's an antibody against PD-1 ligand that suppresses these T cells. And you can easily see that patients that have been treated with placebo, they have a poor survival, about 2 years, and you see there is a significant increase in the survival of these patients when these immune checkpoints are actually inhibited. So today I would like to talk about as we think, a novel immune checkpoint, and that is by when tumor cells produce immunoregulatory glucocorticoids, which then can suppress immune cells. So you all know glucocorticoids, of course. Glucocorticoids are very, very important biologically active ingredients of a lot of anti-inflammatory drugs. Here is a selection, what you get in the pharmacy to treat skin diseases, skin inflammation, other types of inflammation, and so on. But of course, we should not forget that glucocorticoids are actually, or have been discovered as endogenous stress hormones, steroid hormones, produced mostly in the adrenal glands and via a series of enzymatic steps. And what is typical to steroid hormones, that they're produced from cholesterol. Then you have important key enzymes, I just would like to mention here, CYP11A1, because this is the rate-limiting enzyme that really defines how much steroid hormones are produced. And then we have the so-called 11-beta hydroxylase, or the gene is called CYP11B1, and that is the activating enzyme. So these are two very important enzymes. This one drives or defines how much glucocorticoids are produced, and this one defines whether active glucocorticoids are produced. And the active glucocorticoids in rodents are corticosterone, and in humans, cortisol. So glucocorticoids are, for example, induced upon inflammation. So you may have here at your arm, might have a wound that gets inflamed. Bacteria might, get into the skin, and obviously the immune system develops an inflammation. Now, the inflammation may result in the release of, for example, TNF and IL-6, or stimulation directly of nerves, and this triggers the so-called HPA axis, so the hypothalamus, the pituitary gland, steering hormones like ACTH are released, and then the adrenals, they produce glucocorticoids as a consequence of this inflammation. Now, these glucocorticoids, they have directly a regulatory function on the side of inflammation. Actually, for example, if you have mice where you remove the adrenal glands so they cannot produce glucocorticoids, and you infect them with bacteria or viruses, they basically within few days, hours, they actually die because the immune system completely overstimulated and overreactive. So this is a very important feedback mechanism. However, the systemic release of glucocorticoids from the adrenals affects also other organs. For example, the thymus. When you have a strong inflammation elsewhere, the glucocorticoids released by the adrenal glands, they also suppress, for example, the thymus. And this is also why often patients that are under a lot of stress and releasing a lot of glucocorticoids, they become immunosuppressed. So the question we had already a long time ago was the following. Is it possible that, for example, in the intestine, you have a local site of inflammation? This inflammation never gets systemic, and this local inflammation induces locally the synthesis of immunoregulatory glucocorticoids, and these glucocorticoids then provide a local feedback on the inflammation without affecting, the whole systemic system. And in fact, this is the case. We have not been the first one to describe this. It was Jonathan Ashwell from NIH who first described this for the thymus, but we have been fortunate enough to characterize this so-called extra-adrenal glucocorticoid synthesis, first in the intestine, then in the lung, and more recently, we also characterized this in the skin. The idea is the following, that you get some kind of stress, immunological stress, that locally triggers these tissues, and it's noticeable that intestine, lung, and skin, they're all epithelial surfaces, so they're really exposed towards the outside, towards potential pathogens. And these glucocorticoids act then in a paracrine manner. They regulate immune cells and regulate these immune cells locally. And here is a selection of publications that we published during the last 2 decades on this particular subject. So let's have a look how this is actually regulated in the intestinal mucosa. So first, this is actually an experiment we did about 20 years ago, but it still works like this. And what we did is we isolated small intestine. So this is the small intestine of a mouse. You chop it up, you clean it, you make small pieces, and you put it in culture, and you analyze how much glucocorticoids these intestinal tissues actually release by bioassay, radioimmunoassay, etc. And when we first did this, when we cultured intestinal tissue from control mice, we were actually rather disappointed because there was no release of glucocorticoids, or very, very little. However, if we stimulated these mice, when we stimulated the immune system, you can use lipopolysaccharide activating macrophages, or you can use viral infections, or you can use antibodies which activate T cells. Then you get a very strong induction of the glucocorticoid synthesis. And noticeable, this is glucocorticoid synthesis in vitro. And what you also see is if you use a pharmacological inhibitor of CYP11B1, so the activating enzyme, then you see an inhibition. So that we can conclude these are really glucocorticoids which are bona fide produced in the intestinal mucosa, so independent of the adrenal gland. And I'm going to show you more evidence for this genetic evidence. So when we made this discovery, we were, of course, interested what cell types in the intestine are actually producing these glucocorticoids. And at the time, we did not have very good antibodies, so we used so-called in situ hybridization, where you basically have radioactive probes against RNA. And again, we probe here these stereogenic enzymes I mentioned before in the intestinal tissue. And you can appreciate that the signal here in black dots is really at the bottom of the intestinal crypt. So here are the intestinal crypts, and here you see the villi in the small intestine. And this is a very, very interesting location because it's also the location where you have most of the proliferation. But we'll come back to this subject later on. So this is where you have the stem cells or transit amplifying cells, the progenitor cells, and they have a strong proliferative potential. And you're going to see in a second, there is a very interesting correlation between these two aspects. What is the correlation? So early on, excuse me, we got interested in the transcriptional control of this intestinal glucocorticoid synthesis. Now, what has to be mentioned or understood is that in the adrenal glands, there is a nuclear receptor and transcription factor called stereogenic factor 1. So the name is programmed. And you see it's highly expressed in the adrenals. but it's not expressed in the small intestine. And adrenals, for example, they cannot produce glucocorticoids if there is a deletion in SF1. But clearly, this could not account for the intestine because it's not present. However, there is a very, very close homologue of SF1. It's called liver receptor homologue 1, or LRH1. And you see it's not expressed in the adrenals, but it's highly expressed in the intestine. And these are actually the DNA binding domains of SF1 and LRH1. And you see you can basically superimpose them. They actually bind to the same target sequences in the target genes. And how important LRH1 is for the intestinal glucocorticoid is illustrated here. So what we did here, we exposed mice to inflammation, and the mice were either wild type or they had LRH1 deficiency in the intestinal epithelium. And you can clearly see that the inflammation leads to an upregulation of these steroidogenic enzymes I mentioned before here and here. But if there is no LRH1, basically they don't become upregulated. So LRH1 really critically regulates these steroidogenic enzymes. So if LRH1 regulates these enzymes, and these enzymes produce glucocorticoids, and glucocorticoids are anti-inflammatory, well, then the prediction is that mice which don't have this transcription factor are much more prone to intestinal inflammation. And that is really the case. So here you see mouse colonic intestinal epithelium, and we induce inflammation by dextran sodium sulfate. Then this leads to a colitis. And you can see in wild-type animals, you see signs of inflammation, but there is still intact epithelium. Whereas in the LRH1 deficient animals, you see the inflammation is just so strong that this leads to complete erosion of the intestinal mucosa. Basically, all the epithelium here is gone, and you have lots of infiltration of immune cells. So this clearly indicates that this process is really important. So what I have shown you so far is the following. So here you have an intestinal crypt. And I showed you the expression of the steroidogenic enzyme is down here at the bottom of the crypt. And this is also where LRH1 is expressed. And LRH1 regulates the steroidogenesis that obviously has anti-inflammatory functions in the intestinal mucosa. But it's interesting, and I pointed out this location where actually the proliferating cells are. LRH1 has also another function, and that is the regulation of the proliferation of these stem cells and early progenitor cells via the regulation of cyclins that drive the proliferation. And this is very important for the epithelial repair, the epithelial renewal, and so on. But clearly, this function of LRH1 could also be a potential oncogenic function. Now, if there is uncontrolled growth regulated by LRH1, this could lead to tumors. This is actually something we got very, very interested in and that we investigated. What we first did is we actually had a look whether colorectal tumors or tumors derived from these stem cells would express LRH1. And what you see here is tumors or colonic tissue from human patients. And you can clearly see here, or hopefully you can see it here, these brown nuclei. LRH1 has a nuclear staining, has a nuclear localization. These are positive for LRH1, and these are the stem cells of the intestinal mucosa. When we look at the primary tumor, so here in the colon, you see that basically all tumor cells are brown, have brown nuclei, they're all positive, so they overexpress it. And also if you look at metastatic tumors of colorectal tumors in the liver, then you see they're all positive. So obviously, tumor cells have maintained the ability to express LRH1, and potentially could also use it to regulate or produce glucocorticoids. And this is what we were interested in. So here is the experiment with a colorectal tumor cell line, but we can look at this also in primary tumors. And what we did is we downregulated LRH1. So they express LRH1, you will see that later. We downregulated it. And we had a look at cell cycle regulating protein, RB, for example, was not inhibited, was not reduced. But we could detect stereogenic enzymes, CYP11A1, I mentioned, CYP11B1, another one, CYP1117, it's expressed. And more importantly, if there is little LRH1, you see how this is downregulated. So indicating that also in tumor cells, LRH1 regulates the expression of these stereogenic enzymes. Can they produce glucocorticoids? Yes, we find they can produce cortisol. We can inhibit CYP11B1, so the enzyme I mentioned that produces active glucocorticoids. You see the glucocorticoid synthesis is gone. We have here different colorectal tumor cell lines. You see they all produce glucocorticoids. If we overexpress LRH1, we can further boost this. And if we downregulate LRH1, you can see we actually block the glucocorticoid synthesis. So this tells us that also human colorectal tumor cells have maintained the capacity to produce immunoregulatory glucocorticoids in an LRH1 dependent manner. So let's have a look now at real tumors. So not tumor cell lines, but real tumors. And we had a very nice collaboration at the time with a surgeon who removes these tumors, and we received samples from the tumor center, from the tumor edge, which is likely in the most direct contact with the immune system, and also from the normal colonic mucosa. And what you can first see is that, again, these two enzymes, CYP11A1, activating enzyme CYP11B1, you see that it's expressed in tumors at various levels. LRH1 is also expressed in the tumors. And what is really interesting, particularly at this tumor edge, so where basically the normal tissue is in contact with the tumor tissue and potentially also the tumor tissue with immune cells, you see that there's a very high expression of CYP11B1, so this activating enzyme that we will talk about more in the subsequent part of the talk. So tumors express LRH1, and the question is LRH1 involved in tumor development? And if it's involved, is it more relevant for the proliferation of the tumor cells, or is it more relevant for the synthesis of glucocorticoids that then could be kind of an immune escape mechanism? So this is the main question we had. And we decided to address this by using a mouse model of colorectal tumor development. And the model is the following. You inject azoxymetane into mice. This is a carcinogen. It randomly mutates genes, not specific to the intestine, but then you induce intestinal inflammation. And you induce intestinal inflammation by 3 cycles with dextran sodium sulfate that induces only a colonic inflammation, so nowhere else, also not in the small intestine. And as a consequence of this inflammation and the mutagenesis, these mice develop tumors. You can see that here this is the colon, and you see all these tumors here. And this is a model also for ulcerative colitis in human patients that often suffer at later stages from colorectal tumors. So let's have a look how important LRH1 is in this tumor development. Well, first of all, I have to mention we cannot use systemic LRH1 knockout mice because LRH1 is essential for embryogenesis. So what we have to do is we have to use an intestinal knockout for LRH1, so where LRH1 is deleted in the intestinal epithelium. And if you induce tumors in these mice, you see that in the wild-type animals, you have lots of tumors here, and it's very, very obvious that in knockout animals there are much, much less tumors. It's not so much the number of tumors that you see here, but it's mostly the size of the tumor. So the tumor volume, the tumor size is drastically reduced if the tumors cannot express LRH1. Also, when we look at this proliferative aspect of LRH1 that regulates cell cycle, and we look at this by so-called Ki67 staining. So this is an antibody that recognizes a nuclear antigen, a nuclear protein that is expressed predominantly in proliferating cells. So you can see here we have tumor tissue, here we have normal colonic tissue, and you see all these tumor cells are actually positive for Ki67. When we have no LRH1, this is normal colonic mucosa here, but this is tumor tissue here. You see that this is already reduced, indicating that also here LRH1 regulates the proliferation of these tumor cells. So we learned from this experiment LRH1 is critical for this inflammation-induced colorectal tumor development. But what is more important? Is it the role of LRH1 in regulating cell cycle regulating proteins like cyclin D1, E1, c-MYC, etc., and the proliferation leading to tumor development? Or is it more the regulation of steroidogenic enzymes, the glucocorticoid synthesis that then suppresses anti-tumor immune cells and thereby contributes to tumor development? So this is for us was a very important question that we tried to address. And how we try to address this, obviously LRH1 deletion doesn't help much. It's important, but we don't know which direction. This is why we decided to generate a knockout mouse where CYP11B1, so this enzyme here, the activating enzyme, is deleted. What CYP11B1, 11-beta hydroxylase does, it hydroxylases, as the name says, the carbon atom C11 here at this position, and we get from inactive deoxycortisol, we get active cortisol. So obviously a very important enzyme for the availability of biologically active glucocorticoids. And importantly, we deleted this only in the intestinal epithelium. So it's an intestine-specific CYP11B1 using a Willing-Cree deleter mouse, because you cannot generate the CYP11B1 complete knockout, but again, glucocorticoids are very important for the development. So when we generate this mouse, we obviously looked at the synthesis of glucocorticoids under steady state conditions or after stimulating the immune system by using T-cell activating antibodies. And what you notice here in the serum after inflammation, these serum glucocorticoid levels increase. They are not different in the knockout mouse because the adrenal glucocorticoid synthesis is not affected. However, in the small intestine and the large intestine, you can see this glucocorticoid synthesis response is basically abrogated in these mice. So obviously, we have a beautiful model to address the question, how important are intestinal glucocorticoids in the development of colorectal cancer? So if we expose now these mice to a soximatan that mutates genes and then 3 cycles of dextran sodium sulfate, and we just have a look at the weight loss, then we observe something very, very interesting. So the DSS induces inflammation, so the mice lose weight and then they recover again. Then comes the next cycle, they lose weight, and the next cycle they lose weight. And now if you compare the wild type animals to the knockout animals, then you see that the knockout animals, initially they do not so good. They have a stronger inflammation, which makes completely sense. They cannot produce these intestinal glucocorticoids. And obviously, the inflammation is uncontrolled. However, this goes till the second cycle. However, then the third cycle, we see that this phenotype changes, and now the wild type animals are doing actually less good, and the knockout animals are doing much better. And what we did is we took the colon out and we analyzed tumor development at day 35, so before this third cycle and after this third cycle at day 56. Let's have a look what happened. As kind of predicted, these mice here at day 35, the knockout mice, they develop more inflammation. Inflammation really drives tumorigenesis. So not surprisingly, at day 35, these knockout mice have more tumors. However, if you look at day 56, you see the wild type mice, they continue to develop tumors, so the tumor number is actually higher, there are more tumors building up, but now in the knockout animals, there are less tumors, almost indicating if the tumors cannot produce glucocorticoids, then they cannot withstand the attack by the immune system, and they actually going to be reduced in growth and development. So the question is, this is a hypothesis, but how can we actually distinguish between the role of intestinal glucocorticoids during the development or during the inflammation which drives the tumor development, and another role of the glucocorticoid synthesis in the tumor that would actually mediate immune escape and would be beneficial for the tumor, but obviously not beneficial for the host? This is a complex question, and we could obviously not solve this with this colitis model. So what we decided to do is we decided to grow tumor organoids. And I'm pretty sure you heard about this technology. You can, for example, it was built up or developed mostly by Hans Clevers in the Netherlands. You can, for example, isolate intestinal crypts that contain stem cells, and with the right cocktail of growth factors, you can grow these crypts in vitro in 3 dimensions. And you see after 1 day, they start to close, and after 3 days, they start to sprout. So these are basically intestinal crypts that grow, and they grow up to kind of mini guts, if you want to say. So these are really complex organoids with different types of epithelial cells. And you see, if you don't provide them with the right cocktail, they actually start to die. The interesting thing is actually you cannot only grow normal intestinal epithelial cells, but you can isolate tumors from these mice with different genetic backgrounds. You can get single cells, and then you can actually seed them in 3 dimensions, and you can grow them as organoids. And what you see here, day 0, you have only single cells. Day 2, they start already growing as spheroids, and day 4, now they start kind of building balloon-like structures because all the tumor cells have stem cell potential, so they don't form any more crypts and villi and so on, but they just grow and grow and grow. And these organoids we can collect, and we can then inject them subcutaneously into mice, as you see here. So you remove the hair, you inject these tumor organoids subcutaneously, and the beauty is you can, on the left side, you can, for example, inject wild-type tumor organoids. On the right side, you can inject control or knockout tumor organoids, and you can directly compare within the same mouse the growth of these tumors. And you can isolate them, you can measure the size, you can section them, and you can analyze the composition of immune cells, gene expression, and so on. And what you see here, so these are the tumor cells, and then you have lots of stroma, and you see also here you have lots of immune cells, which is obviously very interesting in this context. So if we do so, and we start again with LRH1, the question is LRH1 important? And again, you should now remember that in this context, we don't have any more this inflammation phase that drives the tumor development. We have already tumor cells, but now just allow them to grow, if they can grow, and analyze how important glucocorticoid synthesis is. So what you see, if you transplant wild-type organoids, that they grow fairly fast. You can isolate them, they have built nice tumors, you can section them, you find tumor cells, you find immune cells, and so on. But if you don't express LRH1, but all the other cells in the body, they express LRH1, and they also have a completely normal immune system, you see they are not growing as well as the wild type. And also the tumor size, the tumor weight is different. And actually, if you analyze the histology of these tumors, you see that there is a massive destruction of the tumor cells, but a massive inflammation by immune cells with all these black dots here. So what we then decided was to look not only at the loss of function of LRH1, but actually a gain of function of LRH1. And here it's important to understand that LRH1 regulates the expression of a nuclear receptor, an unconventional nuclear receptor, which has no transcription factor activity. This is called SHIP, or nuclear receptor 0b2. And SHIP is also an inhibitor of LRH1. It binds to LRH1, as you can see here. You can co-precipitate LRH1. You see here, it inhibits the transcriptional activity of LRH1. So we generated tumors from SHIP-deficient mice, and we did the same experiment. We grew them, organoids, and we transplanted them into the mice. And you can easily see that in contrast to LRH1 deficient tumor organoids, we had now a better growth of the SHIP deficient tumors. You see that also here in the size. And also when you do the sectioning, you see much more tumor cells and less immune cells in these transplanted tumors. So far so good. We confirmed again LRH1 is important, but how important now is LRH1 regulate glucocorticoid synthesis in mediating immunosuppression. And this is why we generated, from the few tumors that we got, we generated SHIP-deficient tumor organoids that we also transplanted. And I want to maybe state it immediately, because this was also a question the reviewers had. When you have already few tumors, you might select maybe the strong survivor ones, whatever. We generated also tumor from just floxed mice, so basically wild-type tumors, and then deleted LRH1 in vitro, and we got the same result that I'm explaining now to you. So again, you see here wild-type tumor organoids, they grow subcutaneously, but if the tumors, and only the tumors, have no CYP11B1, they cannot produce glucocorticoids. You see there is a significant reduction in the tumor growth. This is also the case when you analyze the tumor weight, and you can appreciate here when you just look at the size. And what is really interesting, if this was done in immunocompetent mice as recipient, but if you do the same thing in immunodeficient mice that have no B and no T cells, so rag-knockout mice, you see there is still a difference, it's still a significant difference, but it's much more reduced, indicating that T cells, maybe B cells as well, they contribute to the control of these tumors, and they are sensitive to the glucocorticoids. So we then, this is just a glance, we analyzed this in much more detail. We were, of course, interested in what are the cells that we are targeting with these glucocorticoids. And surprisingly, we did not really see major differences in T cells or regulatory T cells. There was no significant difference, but we found a significant difference in the numbers of innate immune cells, particularly granulocytes. What we also have seen is that immunoregulatory cytokines like interleukin-10, which are high in wild-type tumors, they are drastically reduced in the knockout tumors, whereas pro-inflammatory cytokines like interleukin-6 are massively upregulated. And this comes along with reduced expression of so-called M2 macrophage markers. M2 macrophages are known to be supportive for the tumor growth. And you see here that in CYP11B1 knockout tumors, there is a drastic downregulation. So this indicates that really the tumor shapes the tumor microenvironment. So the question is this only the case in this experimental system, or does this also hold true for colorectal tumor patients? And here we had a very nice collaboration with a group in Brazil, Kenneth Golub, and they bioinformatically analyzed patient data that are available in the Kansas Gene Atlas, and they looked for expression of CYP11B1, so the rate-limiting enzyme, A1, CYP11B1, and also SHIP. And what they have seen is that those tumors that express a lot of CYP11B1 or HSD11B1, the patient actually have a poor survival, whereas those that express little of these genes, they have an increased survival. And that was the opposite with SHIP. Remember, SHIP inhibits LRH1. So if there was a lot of SHIP expression, then the patient survived better because LRH1 was better inhibited. And if SHIP was at low expression, then the patient survived less. And this was not the only correlation, but also when we looked at the expression of these immune checkpoint molecules, like PD-1 or PD-1 ligand, we have seen that there was a beautiful correlation between tumors which expressed high or low of these steroidogenic enzymes and the expression of these immune checkpoint molecules. So basically, high capacity for glucocorticoid synthesis permits higher expression of these immune checkpoint molecules and also immunoregulatory cytokines like TGF-beta and IL-10, and that obviously all contributes to immune escape and tumor development. So in summary, I would like to conclude what I've shown you. LRH1 is a very, very important transcriptional regulator of intestinal glucocorticoid synthesis, and this contributes to intestinal homeostasis. But LRH1 in colorectal tumors also controls proliferation and the glucocorticoid synthesis in these tumor cells. And that means that it's equally important for normal homeostasis, but as well as for tumor development. And the glucocorticoid synthesis in the intestine has a dual role. First of all, it controls the inflammation, but at a later stage, it also controls tumor development. And particularly in established tumors, it's important for immune escape. Bruno, do you still have 5 minutes? Okay, I assume we have 5 minutes. So this is basically the model that we have in the normal healthy colon LRH1 regulated renewal of the intestinal epithelium contributes to homeostasis. If there is inflammation, then LRH1 regulates the expression of steroidogenic enzymes. Glucocorticoids are produced, they suppress inflammatory processes and basically contributes to immune homeostasis in the intestine. However, in tumors, when tumors have developed in the colon, tumors can hijack basically the LRH1 regulated expression of steroidogenic enzymes. Tumors can produce then massive amounts of glucocorticoids which impact on tumor infiltrating lymphocytes. They shape also their own microenvironment, for example, by promoting M2 macrophages and so on, and this contributes to immune evasion. And now, of course, a question that you might have is whether this is unique to colorectal tumors or whether this applies also to tumors that are also at epithelial surfaces, for example, in the lung. And actually, what you see here is most recent data where we looked at different lung cancer cell lines, and you can easily see they can produce glucocorticoids. It's blockable by methyl so it's really produced by the tumors. And if you look at the expression of these steroidogenic enzymes in primary lung cancer samples, then you see the rate-limiting enzyme is expressed, the activating enzyme is expressed, and also another activating enzyme are expressed in different types of lung cancer. All right, so with this, I'm at the end. Would like to particularly acknowledge Asma Ahmed, who did all the work in the intestinal tumor development. Verena Merck, somewhere here, that did the work on the lung cancer. And I would also like to acknowledge former people that initiated this work while I was still in Bern. Excellent collaborations with Cristina Jonans. I mentioned Kenneth Collop, Israel Toyota Silva from Brazil, and other colleagues in Konstanz and in Berlin. And I'm happy to take questions that you might have. Bruno, can you still hear me?
Yeah.
There we go. Yeah.
Thank you very much for the talk. Very nice as expected, and also very nice time management. Very sweet of you.
Say it again. I did not understand it properly.
It was also very nice time management. It was very sweet.
Oh, okay. Good. I'm glad for this. Very Swiss. Yeah. Excellent.
But thank you. And what we usually do is that we usually open for questions from the students. And I'm sure if we have, we don't have any on this side. I don't know if Sabo has someone in São Paulo that would like to ask a question.
Mm-hmm.
Yeah, maybe we should start and then just warm up the students.
Yep. Yeah, no problem. I have to escape here a bit because in the seminar room they are building up something. I know it's too loud here. Let's just continue.
It's very nice weather today. It's almost as sunny as here in Rio.
Okay, yeah, here as well. It's actually very warm. Yeah.
But just to get the ball rolling, so I'd like to ask a question that you showed most of the effects on paracrine role of glucocorticoids affecting the immune cells. But do they have an autocrine role on the tumor cells, or even do they have a paracrine role on the normal epithelial cells that are surrounding these tumor nests? And what would that?
Sorry, I'll answer in a second. I just need to ask something. Thomas,
Sure.
ich kann schon noch da weitermachen. Ja. 10 Minuten. Ja. Ja, ich habe keine Verbindung oben im Zimmer. Ich muss da runterkommen. Ja. Okay. Okay, Bruno, I'm with you. I'm answering. So, sorry, I have to escape here. Yes, this is a very important question that you have regarding autocrine functions in tumor cells and epithelial cells. And we don't have yet experimental evidence for this, but it's actually pretty obvious that it will have such effects because, as you may know, the glucocorticoids in the intestine are actually helping to regulate an intact epithelial surface. So glucocorticoids are actually promoting tight junction functions, and anti-inflammatory cytokines like TNF and gamma interferon are actually disrupting the intestinal barrier. And glucocorticoids suppress this. So already this kind of indicates that they have a paracrine function and they're important for this intestinal barrier functions. And the same might actually be true for also the tumors, because particularly in adenomas, they still have like cell-cell interaction. They form like more structured type of tumor tissue, and the interactions might also be regulated by the glucocorticoids. And what we should never forget when you talk about glucocorticoids are the effect on the metabolism. That it regulates glucose availability. A lot of tumors are glycolytic, so they depend on this, and it's pretty clear that this will also contribute to this. Yeah.
Thank you. Next, we have another question. It's that regarding immunotherapy, and showed that therapy, particularly directed against the immune checkpoints regulators, are very important. and glucocorticoids are actually involved in the cell regulation. Is there any efforts to try to put together glucocorticoid inhibitors and immunotherapy to improve the outcome of those treated patients? How are these things going at this point?
Well, I have to say that, of course, these findings that we have, that we just recently published, are extremely new, so it hasn't yet gone into the brains of scientists dealing with these type of questions. But when we have a look at it at a theoretical point of view, I mean, first of all, it has to be mentioned that particularly for colorectal tumor patients, there is often a kind of a adjuvant therapy with glucocorticoids. The reason is, of course, that the chemotherapeutic drugs, they destroy also the intestinal epithelium. That leads to inflammation, and that leads to a lot of side effects which are unpleasant for the tumor patients. However, we don't know whether that potentially has also negative side effects by suppressing the immune system. I mean, it suppresses the inflammation, so it suppresses the immune system. And you know, metabone, what we're using for these experimental approaches, this is actually a drug available for patient treatment for other means. And it could be easily tested for whether, you know, suppressing glucocorticoid synthesis, particularly or preferentially locally in the tumors, would have a beneficial effect, maybe together with chemotherapeutic drugs, etc. Because, I mean, what is also clear is that cancer therapeutics can use cell death. And if this cell death is actually immune cell stimulatory, then this has also beneficial effect. If at the same time you would suppress the glucocorticoid synthesis, then of course it becomes more inflammatory and could be even, you know, breaking tolerance against tumor cells. So I think it has a great potential, but of course the very specific application will be still very, very challenging. Yeah.
Thank you. So I believe we have a question coming from the audience in São Paulo.
Yeah, it's not the audience, it's me, Thomi. Hi.
Gustavo, hello.
Hello again, beautiful work. I have a couple of questions. First is a technical question regarding your colorectal tumor 3D culture, organic culture. Is this only the tumor themselves, or is already a mix of the cells from the gut that you?
Yep. Now the normal cells of the gut, they will not survive. So you initially you might have immune cells, but they will not survive. So eventually what you grow are really tumor cells, and under these conditions. So it's not a co-culture system, but it's purely tumor cells, respectively. If you take intestinal crypts from normal colon, these are normal colonic cells. But the normal colonic cells, they can differentiate into goblet cells, absorptive epithelial cells, and so on. So you get all kinds of different shaded cells in the normal colonic organoids. And who had Claudia had an Gustavo? You had one.
Yeah, no, I'm asking Thomi because we are doing some of these ectopic tumor growth and doing 3D code, so injecting the 3D organoid, tumor organoid, or the 2D, just the mix, the suspension. It gives a different growth rate and things like that. So you don't observe that with your glucocorticoid production?
No, well, we did not specifically analyze it, but maybe they're not highlighted very specifically. What we do is we are not, you know, we are transplanting into congenic mice. They're fully immunocompetent, and this is, of course, very, very important for our study because we really want to see the response of the immune system. This is in complete difference to, for example, transplanting human tumor cell lines into not skid mice where you have no, basically no immune system. But we never addressed whether, the differential growth 2D, 3D would have an impact. For us, it was just very important that we could take the primary tumors and transplant them and then look at the growth.
Okay, and the last question is regarding skin. So you mentioned that also you were studying skin, and we are interested in melanoma, of course. Is there any advance in this your work regarding this, and is any difference between the melanoma and carcinoma, for instance?
Yeah, we this work regarding tumors from the skin. That's mostly from Andrzej Slominski. That's a pathologist in Denver. And he extensively characterized normal sterogenesis in the skin and also in tumors. But what he never did was analyzing how important this is. Instead, we were using the same mouse that I introduced today, but for skin-specific deletion. But we did not analyze it in tumor development, but actually in psoriasis and atopic dermatitis. And it became very clear when you actually inducibly delete CYP11B1, so the skin cannot produce glucocorticoids, the mice actually develop a spontaneous inflammation, almost like a psoriasis-like type of inflammation that is then further boosted if you apply an inflammation model to the skin. So clearly it's very important for skin homeostasis. How important it is for tumor growth, we still would have to test this in a melanoma model or basal cell carcinoma model or something like this. Yeah.
Okay, we actually have a couple of questions. I believe students. The first one, I think, is from Thomi Brunner. I only have the initials here. That's chat, and it was very nice to write down. Nice talk, Dr. Brunner. I have a question about the granulocytic cells. If they have immediate positive increase, could the glucocorticoids be involved on myeloid-derived suppressor cells development?
Yeah, this is a very interesting question, which we did not specifically address, but I told you that we did a much more detailed analysis of the immune cell composition by computational flow cytometry. But this is actually a cell type we did not specifically analyze. But what we have seen is that particularly, as I mentioned, granulocytes, dendritic cells, macrophages, monocyte-derived macrophages, and also NK cells were increased in these mice. And this may actually be somewhat different in this transplantation model versus the original model where you had these 3 cycles of intestinal inflammation. Important point, so we should still analyze these samples, whether these myeloid suppressive cells would also be present in these tumors. Very interesting question. We haven't just having a look. Yeah.
I have another student actually now from Rio, which we know this one.
Hello. Hello, Lucas. How are you? Good to see you.
That was it to you. So I was wondering, we know that in the colon and the intestinal mucosa, there are a lot of mast cells, and it's also known that they express HSD11B1, so in theory they should be able to contribute with steroidogenesis, intestine, even during these processes. And I really don't know a lot about the role of mast cells in colorectal cancer, so I was wondering whether if you investigated and if you know whether they can still contribute even in the deletion of the other steroidogenic enzymes, if they are still important for development of cancer or
actually have to admit that I'm completely ignorant in what's the role of mast cells in particularly in solid tumors. But you know, given that they have overall a tendency anyway to kind of shape the immune system more to a type 2 type of inflammation, and this type 2 type of inflammation is overall beneficial for tumor development. Also, for example, by promoting type 2 macrophages. I would not be surprised if they would not have an important regulatory role in this process, but we never looked at this, I have to admit. Yeah.
I remember I had the impression that you showed an increase in IL-6 gene expression, and of course myself also produce a lot of IL-6. I was wondering if maybe they would be contributing in this regard as well.
Yeah, sure. Yeah. This is you're absolutely correct. This is what we have seen, that IL-6 was increased when we were deleting CYP11B1. Yeah.
Okay. That's it. Thank you.
Right.
Thank you.
We have a question from Claudia.
Yeah. Hi, Thomas.
Hello, good to see you.
Sorry.
Yes. I'm not Claudia, but it's okay. Thomas, I was very curious to know, what do you know about the regulation of SHIP in the malignant tumor cells? I assume probably the expression levels is low in your model, right? But what about in the human tumors that have been isolated, whether this was explored? And regarding the experimental model, is there a chance that by transfecting SHIP into the mouse tumor model, you could see actually a benefit of this transfection? Because it could inspire some ideas about how going about therapy in this case. And whether SHIP is pleiotropic, whether it has, many other targets, or is it relatively narrow? Because
no,
then
yeah.
Okay.
Now, I mean, first of all, SHIP is a direct transcriptional target of LRH1, and at the same time, it's an inhibitor of LRH1, which is already a very interesting connection. But SHIP not only regulates LRH1, but also other nuclear receptors. It actually binds to the ligand binding domain of other nuclear receptors and then kind of inactivates it. The complex thing also about SHIP expression in, for example, tumors or other tissue, it's kind of a dynamic thing. So, it inhibits LRH1, but LRH1 drives its expression, and basically the more active LRH1 is, the more SHIP you get, but the less active is then LRH1. It's a negative feedback loop, which probably depends also a bit on the time point you make this analysis. But regarding your second question, this is actually something we have been extremely interested in, and not in colorectal tumors, but in leukemic T cells, because we have seen LRH1 is also absolutely critical in leukemic T cells, and it there regulates resistance to apoptosis induction. And what we have been trying is to get the tumor cells with inducible expression of SHIP to kind of artificially inhibit LRH1. Unfortunately, this was technically a bit of a problem, but your idea is basically something that we're actively working on to see whether we can use this as a therapeutic approach. Correct, yeah. So hopefully one day we'll succeed. Okay, I see no more questions.
I'm sorry. Close the microphone. We still have one more question here.
Hi, Dr. Brunner. Great talk. And I was wondering, one was more an observation. Showed the T cell infiltrated the person's they're possibly positive cells. But I wonder if you look at CD8 T cells and if they could have been shifted because of also the metabolic moment that the CD8 T cells might be, if they are getting exhausted earlier or later, depending on the of being a regular mouse.
I have to admit I did acoustically not understand your question very well. I just heard something about innate T cells. Is that correct?
As far as CD8, the CD8 T cell, I
okay.
That CD3 positive and CD4 positive,
Yeah.
but I wonder.
Yeah, no, CD8 T cells, I cannot tell you so much about this. I mean, we detect them in the tumor, but I have to admit that, you know, the model system we use with 3 cycles of inflammation, that is different from the transplantation. In the primary model, where you obviously have inflammation-induced tumorigenesis, the situation of the immune cells is much more complex, also because they have normal tissue next to it that is inflamed, and you have the tumor tissue, and it's actually full of T cells. But in the transplanted, subcutaneously transplanted tumors, there seems to be rather a minor contribution of T cells. We see CD8 T cells, we see NKT cells, we see CD4 T cells, we see gamma delta T cells, but there are not really major differences, which does not mean they're not involved in the whole orchestration of the immune system, but we don't see like really obvious changes in T cell levels. Yeah.
Okay, thank you.
You're welcome.
And just you look biogenetically, or were you able to also check their status in terms of metabolic status, or if they were exhausted, or if they were still?
No, unfortunately, we did not have so much possibilities to do this. I mean, I think it will be really interesting also to do single cell RNA sequencing and you know, look for the whole pattern of gene regulation changes in under these conditions. But yeah, we haven't gotten around so far to do this. Hopefully in the future. Yeah.
Thank you very much.
All right, so I actually have one last question, really quick. Is that the transplantation model? It seems in the LRH1 knockout cells, you actually have regression of the tumors. They get they grow and then they start to get smaller. Yeah. But in the CYP11B1, they grow less, but they tend to stabilize in a fashion that resembles the wild type cell. Are these differences real, or just biological variation?
Yeah, I think it's maybe a bit speculative to say it's because of this and this, because these the experiments are quite complex and you cannot do them all in parallel. But when you think about the role of LRH1, that has at least 2 effector mechanisms. One is the regulation of the proliferation. The other one is the regulation of the glucocorticoid synthesis. You would expect that it has a more immediate effect on the growth of the tumor, whereas CYP11B1, the immune system must first recognize that there actually is something wrong which should not be there, and this gives a certain delay, I think, in the response. And from that point of view, it makes sense that the kinetics are not identical for the 2 different knockout tumor organoids. Also, what one we should not forget is, for example, we made sure that these tumor organoids from LRH1 deficient tumors, they still grow. We can still grow them in vitro, but they grow slower. It makes sense. Now it regulates cell cycle regulating genes. And of course, you expect that also the tumor organoids will start growing slower after transplantation. Yeah.
All right, I think that's all for today, and I thank you again very much, Dr. Brunner. It was as exciting as we were expecting it to be. Very nice work. Please tell everybody, your lab, when you go back, that's I send my regards and I miss them. And also Lucas is also send his regards, and it was really very nice to
Thank you.
do today. And we actually we didn't do it as soon as it finished, but you did thank you very much.
Thank you very much to all of you, the entire audience, for joining this seminar. Again, apologies from my side that I could not last time, and also apologies that it was a bit hectic to find a spot in this hotel with the best Wi-Fi, and yeah, but I hope it worked. Good.
All right. Thomi, let's make time this week to chat over the video call, something like that, so we can discuss a couple of things. All right. I have a lecture now, so I have to leave. But many thanks again for beautiful lecture.
Thank you very much.
Okay,
bye.